So it's my great pleasure to introduce Darrel Falk now, whom, as you will have seen from the material in your packs, teaches in a Christian liberal arts college in San Diego. He's Professor and Chair of the Department of Biology at Point Loma, Nazarene University in San Diego. He's a biochemical geneticist, originally from Canada, is that right? And but has moved around the place a little bit. And it's a great pleasure to have him with us today, as you see, to speak about evolution, biological facts and theological challenges, and we heard some of those coming up in the previous talk, so there's another chance to have a second round of questions in a minute. Thanks very much.
Going to be focusing on human
aspects of the biological facts, and of course we're looking at theological challenges from that. This is the year coming up is an important year in evolutionary biology. It represents the 200th anniversary of Charles Darwin's birth and the 150th anniversary of the publication of Origin of Species. And so we'll be hearing a lot, I'm sure, from all different sources as the year progresses. And so I'm going to talk a little bit today about biological facts from the perspective of what we know now, especially focusing in on human genetics and what we know from the evidence as it relates to evolution from the human perspective, human biology perspective. And it's interesting. I come from the States, of course, and there the church folk that I work with, that is the students who come to us at a Christian liberal arts college, most of them have not heard of the idea that maybe evolution, have not even heard actually of the idea that perhaps there's a way of combining Christianity with evolution. Only 28% of evangelicals believe in evolution, evangelicals, and there's 100 million evangelicals in the United States, it's estimated roughly, and only 6% of the evangelicals are estimated to, that's the figure from some polls, as low as 6% of evangelicals believe in natural selection and humans having arisen by natural selection. So the elements that I'm going to be talking about today from the perspective of Darwin, on the one hand, I'm going to argue, but the data is really pretty clear, that on the one hand he was remarkably correct in terms of basic elements of his theory. There's a whole series of facts all these years later that show that he basically had it right. He had a whole series of a set of theological challenges that caused him, in essence, to lose his belief and become an agnostic towards the end of his life, second half of his life, and so we're going to talk a little bit about what his theological challenges are, and I'm going to suggest that what he considered to be weaknesses theologically were just related to his own theology, as opposed to being real challenges that should have been a concern to him. So evolution, biological facts. We'll begin then by talking about the biological facts based upon what we now know about, especially as I've already indicated, what we now know about some things related to human genetics as compared to other organisms, the genes of other organisms. So there's been a revolution, as undoubtedly you know, that has been taking place in the last little while. We're in the process right now of reading the blueprint of a wide variety of organisms, and now have actually hundreds and hundreds of genetic blueprints. In this set of primates, we currently have 2 of them, and chimp and the human, we will soon have all 4 of these. And in addition to that, we're going to have the first draft of the 5th very, very soon. And it's just going to be a first draft. There's undoubtedly going to be gaps in it, but this is the Neanderthal, and we expect some data. There's already data coming out, but a pretty good first draft should be available before very long. And these various primates are in the process right now of being sequenced. Some of them have been completed, and providing us a whole lot of data that we never dreamed that we would be able to have 10 and 15 years ago. And now we have data which is coming in daily that is really affecting our knowledge as it relates to genes and their role in building bodies. I'm going to focus on 3 stories as it relates to the evidence, the evidence of human evolution, just focusing on 3 different aspects that make it pretty clear to biologists that human evolution has occurred.
So a quick little synopsis of some basic genetics that you need as background for some of the things that I'm going to talk about. We've got 46 chromosomes, 2 copies of each one of 23 chromosomes. Each chromosome consists of millions and millions of bases of DNA building blocks. So here's a little portion of this chromosome, and it would just be a DNA molecule stretching from one end to the other. And each building block, each DNA consists of a whole series of building blocks that are called nucleotides. Some portions of the DNA are repeated many times. And so this is just to symbolize a special retrotransposon, it's called. It's given, this particular one I'm going to be focusing on is one called ALU. And so it is found at a particular set of locations. So here, symbolically, at this particular location, there's a segment of DNA that will be found that is actually repeated many, many times and has the capability of moving from one location in the chromosome to another, moving into new locations in the chromosome, I should put it that way. And so this is just the building blocks, the nucleotides of the ALU sequence. And so here we start at the beginning and look at the various building blocks. A little more than 100 or so of these building blocks, and so just reading along GG, CG, CG, all the way through, and that's the sequence of a typical ALU sequence, the one that has the capability of moving into new chromosomal locations. Beginning of a sequence and the end of the sequence, and it fits into a chromosome, and so the sequence would continue from here. And would begin. There'd be a whole set of sequence in here, but this is ALU. And sometimes ALU, when it first goes into a chromosome, well, I mean, not sometimes. When ALU first goes into a chromosome, it causes the chromosome to break. It induces a chromosome break at a particular location, and so here it is in this process, causing a break at that location, going into the chromosome after breaking it, and being inserted into the chromosome. And that's what ALU does: is has the capability of moving into a chromosome. And in so doing, since there's not just one ALU element, there are actually many, many, many copies of ALU, each found at various chromosome locations, each having the capability of moving into new locations. And when they do so, they certainly don't by any means always, but they can damage genes if they go into the middle of a gene, or in various ways can disrupt the function of a gene. And so these are various genetic diseases that are associated with. There's the chromosome in which those diseases are located and found and can be tracked. And what has happened here is that a specific ALU has gone into a chromosome at a specific location, caused a particular disease, such as hemophilia. It's possible to do work with the ALU sequence in Petri dishes. And so here's a set of Petri dishes that have got cells in them, and the cells can be treated with ALU, and you can recover, through some genetic tricks, you can recover cells in which ALU has now moved into a new location. And so it's possible to follow the movement of ALU into chromosome locations outside the body. And when ALU moves into a new chromosomal location, which is what's happening here, one little observation that's very important to our story, and that is the sequence that was pre-existing in the chromosome before ALU had moved into that location. So this is a pre-existing sequence, pre-existing arrangement of bases, and it gets duplicated. And so when ALU moves in, there's now a second copy of a pre-existing sequence, one on one end of the ALU and a second one now on the other end on the boundary, and that's just the way in which ALU inserts. It always generates a duplicated sequence on either end. So with each transposition, ALU is now at a new and very specific location, all different places in the chromosome, and each with their trademark duplication. It's possible to follow the inheritance of ALU since it's moved into a chromosome. Just like a gene, it's possible to follow the inheritance of a particular ALU element. And so in this particular case, then, ALU has moved into a specific location on one of the chromosomes in a male. And what that means then is that the children, 50% about of the children, will inherit that ALU sequence at its new location. Some of them won't. And so this one did not get the chromosome that had the ALU element. This male did, this son didn't, this daughter did, and so on. So you can follow the inheritance of that specific LU sequence down through the generations. In this particular case, this son did not receive the LU at its new location, and so down through the generations, it's not in that lineage. It would be in the other lineages, but not in that lineage, because that son did not inherit the LU at that specific location. Down through the generations, so here's say the great-great-great-great-grandfather. These are just a whole bunch of progeny many generations later, and I'm not showing all the generations. And so maybe this was a couple of hundred years ago in which an LU sequence had been inserted into one of an ancestor's chromosome at a very specific location that can be mapped and sequenced. And here's a child that inherited. Here's a grandchild that inherited, and just not showing all the details. But many generations later, it's possible to look at other individuals and say, well, this has exactly the same insertion. LU is found at exactly the same spot in each of these two individuals. And so these two individuals might now, by this time, be in different parts of the country, different parts of the world, and one wouldn't necessarily even know that they were related to each other. They have a common ancestor back there someplace, except for the fact that you could sequence this portion of their DNA. You can say this person has an LU sequence inserted in the chromosome at exactly the same sequence as that person, and thereby they must have a common ancestor. So even though we don't have that many people here, only 40 or 35 people here, chances are pretty good actually that somewhere back, that some of us undoubtedly have got maybe an LU sequence that might well have been inserted 500 years ago, and we would never have known that we have that common ancestor back there someplace, but we would be able to tell that. We would be able to say, we happen to have our LU sequence inserted, hardly anybody else has it, but you and I have it. That means that somewhere along the line, not too long ago, we had an LU sequence inserted at a particular location. So in this way you can tell people who share common ancestors by the insertion of it. So here's the LU sequence again, which I've just been talking about, and here's a portion. So this is just the LU sequence, and this now is a real human gene, and there's the sequence that is bordering on the LU. So it goes on like this, and so there's sequences of bases, T, A, G, C, C, whatever, all the way here, and then you come to this sequence here, and then the dot just means that LU is there. So I haven't bothered to write it out, but this would be a G here, this would be a G, this would be a G, and so on. There's not quite a perfect consensus with the LU, so it's got an A there instead of a G, but the dot just means virtually everything is there all the way through here, and so it's this human chromosome at this particular location with this sequence has got LU inserted and is running all the way through. And then down here is the rest of the chromosome, which would continue for a long distance. And notice how it has like all LU elements do, it has the trademark duplication. So this would have been a pre-existing sequence that would have been there at the time of insertion. And of course, there's two copies of it now, one on either side, that marks the end of the LU element, and that's just the trademark of an LU having inserted. When it inserts itself, it produces that duplication. So here we have LU now sitting in the chromosome. This special one that I'm talking about, the chromosome is much longer than this, continuing on, but here's the LU, and there's the boundary of it and the duplications associated with that boundary. Now the interesting thing about this particular one that I put on here is that all of us share it. So this is one which we all share. We all have this LU that's inserted at this chromosome location. We all have the duplication at each end, which means then, very interestingly, that means we all have a common ancestor. We all have received this LU element inserted that would have been many generations ago, and it's been passed on faithfully. And since we do all have a common ancestor, we've all inherited this particular one from an ancestor in which it would have been inserted some time ago, a long time ago. So we can ask the question, well, that's interesting, but what about other species that are closely related to us? If we all share it, then at that same chromosomal location, then what about other species that are related to us, like the chimp? And so here's the same chromosomal region. Chromosomal region would, of course, the chromosome continues out here and continues down here, way out into the field there someplace, and here's the LU element in the chromosome, and here's the dots just mean that it's there, it's just bothering to write out all the nucleotides. And the chimp has that exact same LU element few little changes. There's a T here where there's a C here, but it's virtually the same ALU element, and sure enough, the trademark duplication is found at each end. So what has happened is that the chromosome was broken at some point. It's got that duplication, and since it was broken, it means that it's been passed on ever since it was broken. And if this had been two different humans, we would have concluded, well, they share a common ancestor in which the retrotransposon, called ALU, had broken the chromosome at one spot, and we would say, well, we're all related to each other, and indeed, that's exactly what the situation is. But this time, it's not just two different humans, it is two different humans, but it's also, what we specifically were looking at was a chimp and a human, and it's all chimps and all humans that have that same sequence. And so we can ask about what other species, what about other species and other primates, other hominids like us, other great apes, like, and compared to the great apes, and so here's the chimp, and we've already talked about that. Here's the gorilla, here's the orangutan, here's the gibbon, and they all have ALU inserted at exactly the same location. They all have a duplication at each end, and so sure enough, the ones that this whole set of primates have have ALU inserted at exactly that same chromosomal location with a trademark duplication at each end. So we can ask, what about other related species? Like, let's go on to monkeys. And so if we go on to monkeys, like for example the vervet, then we can look at that and we can say, well, here's the equivalent chromosome region. So it's definitely all down here would be the equivalent chromosome region, the similar sequence, similar genes, and it would continue on down here. And we can say, well, at that chromosomal location is ALU present, and the red dashes that doesn't mean it's present. In this case, it means it's absent. So there is no ALU in that region at all. There's nothing there. And so here's the pre-existing sequence, and it's not present at the other end. There's only one copy of it. So what that implies then is that there was an insertion. And the insertion probably occurred quite a long time ago, because it's present in humans, it's present in chimps, it's present in gorillas, present in orangutans, present in gibbons, not present in monkeys, and not present in other of these primates, the ones that have been checked. So what that implies then is that this, somewhere back here, that means about 18 million years ago, somewhere back here, ALU was inserted into a chromosome in a particular location, and it's been passed on faithfully, generation after generation after generation, for the past 18 million years. All of those organisms that had this common ancestor in which the event occurred have been passing on this insertion faithfully all through the generations. But because Old and New World monkeys don't have that ancestor, they come from a different lineage. The lineage broke up in here someplace, and so they don't have it, and none of these other ones have it, of the ones that have been checked. So that's pretty strong evidence then that that ALU is a way of tracking. It's a pretty strong evidence of the common ancestry of these various primates. Altogether, there's not just one ALU element. I've been focusing on one, but there's not one. There's actually over 1 million of them, and you can follow these sorts of things for a whole set of them. And by examining which organisms share the insertion, you can tell about when the insertion occurred. And so sometimes for certain ALU elements, the chimps and the humans would share it, or in others it might be the orangutan, or say the gorilla and the human and the chimpanzee that would share it. It'll be interesting when we get the data on the Neanderthal to fit that into the story as well, because in part 7,000 insertions have occurred in here since in the time, about 7 million years or so ago, since chimps and humans had a common ancestor. There's been insertions along the way, and ALU has been inserted in about 7,000 unique insertions are found in one species or the other, but not found in both. So ALU continues to be inserted. And we need to remember that this is something that has been documented to occur in petri dishes, and we can tell when it's happened in petri dishes. It's got the duplication at each end, and that's what we find when we look at them, when we look at ALU in the organisms themselves. So we can ask the question, gradual creation in humans. there are many cases when exactly the same insertion of a transposon, just a quick little review, the precise spot is shared by people who are related to each other. It's because hundreds of years ago an insertion occurred in an ancestor they all share. There are many cases where the exact same insertion of a retrotransposon, LU being one example of a retrotransposon, there's others, into a precise shared spot is shared by humans and chimpanzees. Almost all geneticists are certain that this is because the insertion occurred in an ancestor that they both share. And so the question is, if God created humans, and you can imagine, especially for me to give this talk, which I have a number of times in America, where most of the evangelicals that I speak with are pretty uncomfortable with this, that this is a concern. And so I asked this question in this fashion: if God created humans by a gradual process over a long period of time, would this somehow make it less God's creation? Here's my second story that I want to talk about as we work our way through evidence as it relates to Darwin having basically had his biological facts right as it relates to the story of human creation. And I'm going to consider chromosome 2 at this point, one of the human chromosomes. There's 23 pairs of chromosomes in essence, and a little difference with respect to the X and the males have an X and a Y chromosome. I won't go into that, but it's in essence 23 pairs of chromosomes. And the same genetic information is present on 2 different chromosomes in gorillas and chimps. So this is a gorilla, 2 of the gorilla chromosomes. One's called chromosome 12, one's called chromosome 11. Here are 2 chimp chromosomes. One's called 13, one's 12. They have virtually the same exact genetic information on them all the way through. The bands, the dark oranges, they correspond to stains. It's possible to stain the chromosomes, and you can actually see just by the banding pattern of the chromosomes that they're equivalent, the same, and gene by gene, even nucleotide by nucleotide, they're very, very close to each other and have the same genetic composition. And but in humans, it's interesting. The bands are there, the genes are there, the DNA sequence is there, but it's all found on 1 chromosome and not 2. And so they don't exist as 2 chromosomes in the case of humans. Humans have 1 less chromosome as a result, and so humans have 23 pairs of chromosomes in a gamete, 23 chromosomes in a gamete, and chimps and gorillas and orangutans have 24, because of what people think was a gene fusion event, and that gene fusion event resulted in 2 chromosomes that in the ancestor had been separate from each other, but now became joined to each other to become a single chromosome. And it's more than just the coincidence of the 2 chromosomes having the same genetic information here, but now having all the information on 1 chromosome here. It's more than just that. There's another part to this story which is especially interesting. At the end of a chromosome is a special sequence known as a telomere. And so a telomere is a special sequence that is found at the end of a chromosome and needs to be at the end of a chromosome. It turns out that this telomere sequence is important for replicating a chromosome. It won't get replicated properly. We won't go into the details as to why it's not important to our story, but there needs to be a special sequence, a special arrangement of the nucleotides, a special order of the nucleotides found at the end of a chromosome. And if a chromosome doesn't have that special sequence, it's not going to get replicated properly. I'll say something about this red area called the centromere in just a minute. Okay, so here's what people think happened at the time of fusion. That would have brought 2 centromeres together. Sorry, 2 telomeres together. And so you can now look, and indeed this has been done, you can now sequence the chromosome and ask the question, what about the 2 telomeres that are supposed to only be at the end? And what you find in this chromosome is a telomere sequence that has no longer has that old function, but it's still there. It's mutated a little bit. The sequence is not a perfect match. The 2 sequences are in inverse order. It's very clear that they used to be part of the end of a chromosome, and so what that means to virtually all biologists without question is that there was a time when there was an ancestor to human beings in which this chromosome existed as 2 distinct chromosomes that got joined together, and still residual to that, we have the telomere still there, but no longer functional and indeed mutating out of existence. The other important part of this story, and that's the centromere, and so chromosomes have centromeres which are important in dividing of chromosomes, and so each chromosome needs to have a centromere. And the reason they have to have a centromere is that the centromere is important in attaching to a little piece of string. We can think of it that way, that piece of thread made out of protein, a protein that plays a role in pulling the chromosomes apart, and the two duplicated parts are being pulled into two new cells. And so that they need to have a centromere for the string to attach to pull the two duplicate halves in to make two new cells. So that's what's going on here, a process called mitosis. And so every chromosome is going to have a centromere because it's going to have to get attached to the string. And so here, if we look at this, we know there was a centromere here in the gorilla, and there is a centromere here in the case of the gorilla and the chimp and other organisms as well. And yet, if we look at this chromosome, what we find is the sequence is still very similar to a centromere, but it's mutated and it's changed. And so what it is an old residual centromere. And so clearly what virtually all biologists would say is that there was a time when these chromosomes were in an ancestor were two chromosomes, two distinct chromosomes. What's happened is they got joined together, and the old centromere is now disappearing. And as time goes by, it's accumulating mutations, but it's still there as a trademark of that previous event. So this is pretty strong evidence then that there was a common ancestor in which the two chromosomes got joined together, all of the great apes have them, and humans are joined together, and we've got all the residual trademarks of that event still being present in the chromosomes. So we can ask the question, could it be that God guided creation to go in the direction it's gone, but has done it through a gradual process? And the answer, of course, based on this, is that this is exactly what one would expect. This sort of thing is if God had created humans through a gradual process of this sort from previously created species. And so that's evidence, story number 2. I'm going to talk about story number 3 now, which relates to something which is known as synonymous and non-synonymous mutations. I'm going to explain what that means. As we proceed, it's related to changes in the DNA, particular kinds of mutations, that we're going to compare some DNA between humans and chimps as we do so. Before I can get into that, though, I need to tell you just a little bit about what DNA does and what genes do. So here's a DNA molecule, a little tiny portion of a DNA molecule. It'll be much, much bigger than this. That consists of 15 or so of the millions of bases that would be in a single chromosome. But as far as what a chromosome does, what DNA does is it's in the form of a code. And so what we have are 3 letters of the DNA, 3 nucleotides, 3 building blocks of the DNA, each specifying a particular amino acid. So this set of nucleotides here says, in building a protein, so this is the product of a gene, and this is the building blocks of the protein. They're known as amino acids. And so each portion of the gene, each portion of a DNA molecule says, plug in building block serine, SER. And then this portion says, okay, now plug in the building block aspartic acid. And this portion says, all right, now plug in the building block PHE, phenylalanine, and so on. And that's in essence what is done in building the product of a gene. And so all of our different proteins are built in this way, reading the code, because there's a code in the DNA, reading the code, making a protein, and that order of the amino acids is very, very important. It allows the protein to fold up in the way that it normally folds up so it can carry out its work. And I'll show you an example. So when everything is, when the gene is built of protein, it will consist of not just 8 amino acids, 8 building blocks. It will consist of many, many more than that. Perhaps a couple hundred, depending on the protein, and that will be the gene product. And it'll then be able to take on its particular shape. And this is an example of one protein, the protein hemoglobin, which we're familiar with. And there's a gene, a couple of genes that are involved in making this protein. And it's a chain of amino acids that simply fold up on themselves. Several chains of amino acids that fold up on themselves. So I just want to look at one particular example now of a DNA, a gene, portion of a gene, the gene that specifically makes a portion of the hemoglobin molecule. And so this is the DNA sequence that we've been looking at a little bit, the nucleotide sequence, which is in the form of a code. and GAG specifies or codes for GLU. And so there's a gene that we all have, and somewhere partway through that gene is the GAG, which says plug in GLU, plug in glutamine here, or glutamic acid here, and so it does. And then the next one says plug in glutamic acid, and so on, all the way through the whole protein. And when everything is all done, although we just look at a little portion of the protein, a little portion of the gene, we get a protein molecule, hemoglobin. Actually, within the cell, we have millions of them, and so here's a red blood cell with with millions and millions of, there's billions of hemoglobin molecules inside of it, each product of a gene, which has specified the amino acid chains and how they fold up in this fashion. However, when mutant, when the gene is changed, which we have here, instead of having an A at this location, there's been a change in the instructions. Mutation has occurred. And so instead of having an A like you're supposed to have, now you have a T in there, and a T doesn't say to plug in glutamic acid, it says plug in valine. That's the wrong amino acid. So as a result, when the protein folds up, it folds up in the wrong way. It actually folds up more or less correctly, but except for one thing, and that is that it forms these long strings. Instead of the protein molecules being somewhat independent of each other, it's just a long conglomerate of protein molecules, and that causes big problems for the cell to have these long strings of stuck together protein molecules, and the cell folds abnormally, takes on an abnormal shape. So this is a red blood cell that's not shaped like it should be, and it keeps getting stuck in that abnormal shape results in it getting stuck in the tiniest of the blood vessels, blocking the blood flow, causing a great deal of pain, and then eventually death. Mutations which change the sequence of amino acids are called non-synonymous mutations. So the one that I've just been talking about, where the sequence was changed, that's called, and the wrong amino acid is plugged in, that's called a non-synonymous mutation. Let's just see how a non-synonymous mutation is generated. We're going to look at the genetic code just briefly, so you can kind of get a feeling for how it works. And so here's TCT, which specifies one of the amino acids, serine. And there's other codes, like for example, if it's TTT, then that specifies phenylalanine. Different amino acid. If it's TAT, that specifies another building block, and TGT specifies another building block. And so if we have TCT, which is supposed to specify serine, and then that C changes to a G, then what that means is that instead of serine being inserted into the protein product of the gene, we have cysteine inserted into the protein product of the gene, and that's what we call a non-synonymous change. It's not the same. It's not synonymous. It's a different protein that's being produced. There's a whole different class of mutations which are called synonymous mutations. In this case, as it implies, they are the same, and the reason for that is because you can have more than one way of building serine. So TCT specifies serine, as does TCC, and as does TCA, and as does TCG. They all specify serine, and there's a couple more as well. So here's the code as a whole, which we obviously won't worry about, but I'm just trying to say that this is all part of the genetic code. And sometimes there's redundancy in the code, so you can get the same code specifying, different codes specifying the same amino acid. So in this particular case, we would have the sequence, the codon will code TCT specifying serine, and a mutation might occur where that T is changed to an A, but in this particular case, it doesn't change the amino acid. It's still serine that's inserted, and we would call that a synonymous mutation. So there's the two different types. We know that they occur at the same frequency. There's all sorts of data from the last 50 years which indicates that synonymous and non-synonymous mutations occur, and they're produced at an equal frequency. However, since non-synonymous mutations frequently produce defective proteins, they don't survive with equal frequency. So if there was a common ancestor a long time ago, and if there was a branch that occurred since that common ancestor, and there's been mutations occurring in this lineage of the branch, and there's been mutations that occurred in this lineage of the branch, given that synonymous mutations survive as the lineage, here's the common ancestor back here, given that the synonymous mutations survive, if you look at this lineage, you might find lots of synonymous mutations that are found in both lineages because they're tolerated. synonymous mutations would be tolerated here. They'd be tolerated here, so you might find lots of synonymous mutations that have occurred simply because they're tolerated. On the other hand, let's say that down here we have a non-synonymous mutation which changes the amino acid sequence. That one would probably result in lethality, because that would be lost to the lineage. Non-synonymous mutation changes, like sickle cell anemia, changes the amino acid sequence in a way which is detrimental, and so you wouldn't find non-synonymous mutations. If indeed there was a common ancestor and we've had these two lineages, then the synonymous ones would be passed on faithfully in both lineages. They'd be passed on faithfully generation after generation. The non-synonymous ones would disappear. So synonymous mutations can be tolerated. Synonymous mutation here would be tolerated. A non-synonymous mutation would not be, as well, anyways. So God created humans and chimps through a common ancestor. We would see lots of evidence for surviving synonymous mutations that are different between the two lineages. Okay, so synonymous, I'm not sure I quite said that right. I can't remember exactly what I said, but I need to make sure if a synonymous mutation occurs here, it would be tolerated and go on through, and wouldn't be found over here. That's what I hope I said. Okay. Since many non-synonymous mutations will have deleterious effect, if a synonymous mutation occurs here in the time since they had a common ancestor, then that would quite possibly be lost. All right, so what we have now is the sequence of 13,000. We have the sequence of the whole human and chimp genome, but a comparison was made between between 13,554 genes in the human as compared to the chimps, and so we can look at those genes, and indeed at 5,500,000 sites, and when you do that, when you compare the 13,000 genes at 5,500,000 building blocks in the human compared to the chimp, you find that this is for protein coding portions of the DNA. You find that there was 1.23% difference according to this Nature paper that came out 3 years ago. Now here's the important point to us, and that is, sure enough, the rate of tolerance of even though we know they occur at an equal frequency, there's lots and lots of data to show that they occur at equal frequency. If you compare the two, there's not very many non-synonymous mutations that have occurred, so that means then, I mean, they still are tolerated sometimes, but there's 1/4 as many. And what that means is that quite frequently when a non-synonymous mutation occurs in a lineage, it results in death, and it's not found. So when you're looking at the differences between the two, you don't see very many non-synonymous, or at least 1/4 of the events that are non-synonymous compared to the number that are synonymous. And so this then indicates that it's exactly what one expected. Chimps and humans were created from a common ancestor. Changing the amino acid sequence is frequently deleterious. So since both types of mutations occur at the same rate, could it be that they have a common ancestor and the non-synonymous mutations don't get passed on to subsequent generations as effectively? I think I've got time to just talk about one more aspect, and then I've got some other things that I need to say about theological challenges. So this is still synonymous and non-synonymous. I just want to say a few more things about genes, though. And but this just provides further data as it relates to that. So genes are divided up into portions which code for a portion of a protein. But there's also sections of genes, we didn't talk about this, but there's sections of genes which are known as introns that don't specify a part of the protein. They actually, they are not used to build a protein. And so genes are split up into regions which are coding, that is, specify, provide the information how to make a protein, but within those genes also are some sections which don't specify protein. So this is then especially interesting because you would expect that if this is not involved in making a protein, that we probably could change it and it would be tolerated. And so let's take a look at data as it relates to that. So here we go. This is mutation rate within the coding portion, so changes that occur within the exon, as it's called, the coding portion of a gene. This is the portion in the coding portion which does not change, so this is synonymous. This is synonymous, doesn't change the amino acid sequence. These are those which do, and so these are both within the exon region, but these are changes that change the amino acid sequence. These are so non-synonymous. Those are synonymous changes. So that's what I've already talked about. Now I move to the next part. Well, if they're in the non-coding region, actually they're tolerated even more effectively. There's some special reasons for that which we won't go into, why they are tolerated even better than synonymous mutations, but that's another story for another time. And then finally, if they're right at the junction, interestingly enough, right here, this sequence has to match up perfectly, because if it's not a good sequence, then you're not going to be able to tell where the junction between coding and non-coding is. We know all the reasons for that. And so this sequence has got to be exact. And you hardly ever get mutations if you're comparing the two lineages, chimps, humans. If there's a mutation that occurs at that junction, it's virtually always wiped out, apparently. So more evidence then that for a common ancestry. And I think this was about as close. There's other kinds of data, which I'm not talking about, paleontological data, which of course is extremely strong. But this is about as close as you can ever get to a proof in biology as it relates to human evolution. And the data is extremely strong to show that the data indicates that God did create humans in a gradual fashion. Mutations have occurred in life's history, just like they have occurred today. Over millions of years, individuals that carry deleterious mutations, in this case non-synonymous, exon-intron boundaries die early or reproduce less effectively over millions of years. Ancestors that carry mutations that are synonymous, the introns are seldom negatively impacted, and thereby they continue the lineage. All right, so with that then as background, that's the biological facts portion that leads to strong evidence that actually Darwin had things right as it relates to the story of life. But he reached the conclusion that the theological challenges associated with it meant that he needed to abandon his faith. And I'd like to spend my remaining 20 minutes just talking a little bit about the theological challenges that he posed that caused him to lose his faith, because of course he began his life began his, just prior to going on the Beagle on his voyage, was planning on being a minister, and he went, I think he studied right here at Cambridge, to prepare to be a minister, and then along the journey, and in the aftermath especially of the journey, he lost his faith. It was a long process. We heard a little bit about that this morning. I don't mean to say that it all happened on the journey. In fact, it was a lifelong process in some ways, different aspects of it. And he did changes, he was back and forth, as we heard this morning a little bit. But in essence, he became an agnostic. And became an agnostic more or less, probably within a year or so of the time he got back from his journey, right at the time he was formulating the theory of natural selection. All right. And for him, for Darwin, there are several things that he was deeply concerned about. One of the biggest issues for him was the incongruence between and we talked about this actually last night a little bit, the incongruence between a God who is benevolent, omniscient, all-powerful, the kinds of questions that we actually talked about last night, and they came up again today as well. So there's that image of God as being all-loving, and yet for Darwin, then there was this question of his discovery that new life arises through pain and suffering and competition for survival. And so he had on the one hand this view of God that was all-loving and all-powerful too, so he should have been able to do it in a better way. And yet this is what Darwin discovered. It's pain, suffering, all the way. It's competition, all the way. This is a little too close. But. So he came to believe that his theory was the death knell to believe in a God of that sort. So, of course, that's a big part of why he kept it quiet for 20 years. Was he had reached the conclusion that he had discovered something that had every reason to shake up people's faith. And he was very concerned about that. There were other reasons as well, but that was certainly one of the big ones. So this is what he wrote. For example, 1856, what a book a devil's chaplain might write on the clumsy, wasteful, blundering, low and horridly cruel works of nature. I cannot persuade myself that a beneficent, omnipotent God would have designedly created the Ichneumonidae with the express intention of their feeding within the living bodies of caterpillars, or that a cat should play with mice. So I couldn't picture God working that way. not a loving God, not an all-powerful God. Why would God do it in such mean ways? It's important, I think, that we see that there's nothing particularly new about the challenge that confronted Darwin. We live, as we've been talking about, we live in a world of pain and suffering. It should not surprise us, and probably should not have surprised Darwin, that the story of creation is interwoven with the story of suffering. From the human perspective, all of us would like to remove suffering from that story of creation, and we're still struggling with it, just as Darwin did. We'd even like to remove suffering from our story. We have a whole book of the Bible, actually, that, as we all know, that focuses exclusively on the problem of pain and suffering, and devotes 42 chapters to it. Of course, that's the book of Job. And for 37 chapters, back and forth in discussions and so on, Job cries out to God, in essence, and others as well, in distress and anger, of course, about the apparent injustice of suffering. In the 38th chapter, Job begins to listen. The climax of the book comes when Job is finally confronted with his own human naivete, in light of the mystery of God and God's ways in the universe. And Job, in a very real sense, speaks for all of us as believers, really through the ages, when this is the conclusion he writes as it all comes to a close. Surely I spoke of things, he says, I did not understand, things too wonderful to me. My ears have heard of you, but now my eyes have seen you. So, what did his eyes see?
Well, if you take your name tag off, I think you'll get rid of that. Your name tag keeps hitting the mic. That is my thesis.
I bet you're right. All right. So, anyways, what did he see?
No, you should share.
I'll put it down here. So anyways, what did he see? Didn't he just simply see God's presence? I don't mean that he saw God with his eyes. I think he recognized God's presence in the universe. And I think his mind simply came to trust in that presence, despite his inability to comprehend the problem of pain and suffering. So I think what happened was that he came to trust in that presence and the mystery that was associated with it, and he came to trust in God. Perhaps from the theological perspective, Darwin was really just asking the question of the ages. Perhaps from the theological perspective, Darwin had not unlocked a new question at all. As Darwin completed his theory of evolution, perhaps the issue was that Darwin, unlike Job, never made it to the 38th chapter, where he listened to God as God spoke to him. We just can't remove suffering from the world. Indeed, when we soft pedal, when we romanticize creation as being the work of an engineer, rather than the work of a God who builds freedom into creation, with all of the randomness that might imply, messiness anyways, we point to a reality that just doesn't exist. So, as I see it, if there's any one message to the entire ongoing saga of the interaction between God and the Hebrew people, for example, the 40 years wandering in the wilderness, it's that God chooses to create through an interplay. God chooses to create through an interplay between freedom and necessity. And when we fail to recognize this in our own lives, when we fail to recognize this in the story of creation, we become responsible, I think, for perpetuating Darwin's dilemma on into 150 years now, on into the 21st century. So, has our understanding of suffering and pain progressed much beyond where it was left in the 42nd chapter of Job?
2,500 years ago? Based on our discussion of last evening, I felt the answer is no. John Polkinghorst gave us some interesting things to think about today. But nonetheless, probably not that much real progress has been made 2,500 years later. It's still a mystery, and maybe that's where God wants it. I just said maybe. The answer to the problem of pain and suffering is hidden somewhere in the mystery between a God who wills freedom for creation and a God who is sovereign, somewhere in the middle there. A God who Job, at least, finally came to see. God who Job finally came to see really knows and understands infinitely more than we do, and infinitely more than we can ever hope to know and understand ourselves. So, in the midst of pain and suffering, however, beauty keeps peeking through. The world, of course, is incredibly beautiful, but that beauty has come about through pain and suffering, pain and turmoil. The story of creation, as Darwin discovered, is no different than the world that we see as bad things happen to those that we love. We never get an answer, really, to the question of why it works this way. But through hope, we keep getting these glimpses of beauty. And in those glimpses, every now and then, we're surprised by joy. And God's Spirit is at work in us as we struggle against the chaos. And we can focus on the Spirit and what emerges from the chaos, or we can focus on the chaos and lose sight of the Spirit that's at work. And that choice is ours. Darwin, of course, chose to focus on the chaotic forces and assumed there was no Spirit at work. As believers, we focus on the Spirit, on God's Spirit, and what God wants to bring out of the chaos created by freedom. Like Job, we don't really understand the details, but in faith, we assume that there is no other way. So that's the first challenge that Darwin faced, and I guess my point really is simply I'm not sure that was a new challenge. And it wasn't anywhere near the challenge that people have been faced many, many times before, and we still have to face today. Second theological challenge, which deeply impacted Darwin, related to the fact that he was deeply steeped in the thinking of William Paley and his design engineer, his watchmaker hypothesis, where I'm sure you're all familiar with it, where there was if you discovered a watch on the beach and you picked it up and looked at it and the way it was working forensially, you would know that there was somebody that had designed that watch. And of course, William Paley, being a pre-ministerial student here, was deeply steeped in that. In fact, he was a great admirer of William Paley, and indeed, while he was a student here, resided in the very same room that Paley had resided in some years before. So, given this built into his thinking, as he was, we find these kinds of things in his diary as he was on his voyage. Among the scenes, Darwin wrote, which are deeply impressed on my mind, none exceed in sublimity the primeval forest undefaced by the hand of man, whether those of Brazil, where the powers of life are predominant, or those of Tierra del Fuego, where death and decay prevail. Both are temples filled with the very production of the God nature. No one can stand unmoved in these solitudes without feeling that there is more in man than the mere breath of his body. No one can stand unmoved in these solitudes without feeling there's more to man than the mere breath of his body. I especially like this quote. I felt glad I was by myself, reflecting on a magnificent scene. It was like watching a thunderstorm.
for hearing the full orchestra, a chorus of the Messiah. Here's Darwin, still as a believer on the trip, maybe beginning to change. It's actually not clear from those diaries when the change took place and when he really came to believe in evolution. Most scholars think it didn't happen until the trip was over with. 5 years. It was a 5-year trip. Most scholars think that he didn't really come to believe in evolution until it was over with. There are some that think that it was changing, but there's very little sign from the diaries. Anyways, as the years went on, he came to see that the apparent random forces were at work in the natural world, and it caused his view of a watchmaker God to come crashing down and fall into broken, random pieces. His image of God was gone. His view of God as a designed watchmaker was gone. This is another issue, though it's not new. This notion of randomness, and the ancient Hebrews thought about this, of course. Question of God's presence in the in the midst of randomness, and so we've got various portions where you can just see this kind of thing going on. Like, for example, the story of Joseph is a great example, I think. And so here we have this story of Joseph being tossed into a pit, and just as the Egyptian slave traders come by and just happen to come by, and then Joseph ends up in Egypt and meets up with someone who has connections to Pharaoh, and all that just happens that occur in that, with Joseph eventually becoming chief assistant to Pharaoh, and that causes Joseph to say this: "You intended to harm me, but God intended it for good, to accomplish what is now being done, the saving of many lives." So the story of Joseph is the story of providence through this series of events that somebody looking on from the outside might look and say, that was lucky that he happened to meet with him, or that was a good fortune that that person who was going to someday hear Pharaoh's dream, that he would meet up with him, and all those different events. And yet here he is saying, you intended to harm me, but God intended it for good. In other words, God was involved in all this, what some people might interpret as being a set of random events. So the issue of providence in the midst of seemingly random events didn't arise at all for the very first time with Darwin. In fact, built into the wandering in the desert, all the different events that we see in the Old Testament are built into that. I mean, the events seem to be, somebody looking from the outside wouldn't necessarily see God at work, event by event. And so anyways, it seems to me at least that this is in a very real sense something which people were thinking about way back then. And even today, in the 21st century, each of us looks at the events of our own lives. The college that we happen to choose to go to, and how our lives went in a particular direction. Would have gone in a very different direction, perhaps, if we hadn't chosen to go to that college. Or the chance meeting with somebody who eventually became a spouse, for example. Or the job we got, and how different our lives were because we got that job. Or the job we didn't get, and how different our lives are because we didn't get that job. And so we have all of these events taking place in our life, and some of them are things which can cause everything to change. And to anyone looking from the outside at the events of your life or the events of my life, they might look at those events and say it's just a whole series of random events, or a whole series of events that, I mean, not necessarily totally random, but just chance meetings, various random events. And yet, for those of us as believers, we happen to believe that God has been guiding the presence of our lives in various ways. The story of belief is a story of a belief in God working through the events in our lives. Some people, of course, believe that God is at work in every single event in our life. I can think of a book that became a bestseller in the United States, where the author really did indicate every single event God is happening in some preordained fashion. Other people believe that it's not that every single event was preordained, and indeed some people would even say everything really is somewhat random. Everything really is random, and God doesn't intervene in the events of our lives. There's plenty of people that believe that, but all of us as believers believe that God's presence is real, and that God's presence is there in the midst of that. So the problem with Darwin, that Darwin brings up, as I see it anyways, and the seemingly randomness of life's history, is not a new problem. And the fact that he thought it was is indicative of the inadequacies of the theology of his day. Indeed, the fact that some continue to be troubled by this, God working through apparent randomness, today means that it's kind of futile to try to sort out what is random and what is not random. And there are groups of people, especially in the United States, that are trying very hard to use mathematical calculations to show the probability that all is not random with respect to, and kind of answer Darwin's question of randomness. I would suggest that that's not possible, to be able to prove, just like I can't prove the events in my life, what's random and what's not random. For example, when I was in London a few days ago, I had this impression that I should button the back of my wallet, my back pocket here. Okay, so I did. I don't know if I've ever done that before. I must have done it somewhere along the line, but I hardly ever done that. I should have done it many times, of course, but I didn't. And I never had to before. So I had this distinct impression. Well, you know what happened 3 hours later, as I was walking along Russell Square. I heard this rushing upward behind me, and reaching in my pocket and grabbing and trying his best to pull out my wallet, and couldn't get it. I told my wife about this, and I said, I think it was Providence that I remembered to button my wallet just before this happened. And she said, Darrel, do you remember the last thing I said to you? I said, don't get mugged. I would maintain that God is at work in our lives. And it's not possible through calculations of some sort to figure out where God was involved and where God's not involved. But as I look back at the history of my life, and as you look back at the history of your life, I think the issue is that God's presence is always there. And God is always guiding our lives. God's at work guiding our lives. And although we probably can't ever come down to the point where we can say, this is where God was involved, this is where God was involved, and so on, through some sort of mathematical calculations, we don't believe that all is random in our lives. We believe there's a guiding presence in our lives. And I would say the same thing then with respect to what's happened in creation. We stand back and we look at the beauty of creation. We look at the beauty of the lives of people that I know who are believers. And I look at those lives and I say, and in a very real sense, I'm drawn into worship by the beauty I see in their lives. So also with respect to creation. A whole series of events have taken place in a world that God created in freedom. Can I go in there and say, step by step by step, here's where, and I'd say, no, absolutely not. That's my personal opinion. But on the other hand, I stand in awe of the end product. I, in essence, bow down on my knees in worship at the beauty of creation. Thanks very much.
Thanks very much. Perhaps I'll just take the privilege of being chairman to ask a technical question from the first part of your talk. And I'm just a geologist, so I'm just a simple man with rocks. But when you showed the exons and the introns, I thought you were going to say that the number of non-synonymous duplications was more than the synonymous ones in the bit of the genome that isn't used there. That's the introns, isn't it? But you didn't show that diagram. Is that true or is that not true?
So the number of non-synonymous mutations in the exon portion?
In the bit that's not used, whichever that's called, is that the intron? I would have expected that to be more, because it doesn't matter, so they just mutate. Is that the case or not?
Well, since all we have is the sequence itself, so it's not coding for anything, so you can't really say here's a synonymous versus non-synonymous. It's just not coding. And so one can't really make that distinction. This is coding. This is the coding portion of an intron. This is the non-coding portion of an intron. So you can't really say synonymous versus non-synonymous. It's all non-coding. So what you do have is exactly what you're saying, and that is the whole thing is more. It's more random.
So you can't identify CGT and say that's a coding portion that's no longer used.
No, it makes no sense in the sense that there's different reading frames, and so you can't say this is the equivalent. Yeah.
Because you don't know where to start the sequences. Right?
Exactly. That's it.
Thank you. Let's have some proper questions then.
I'm an evangelical Christian, I'm also a scientist, and so I don't have any trouble with the gradualism story as you've been explaining of evolution. If God wants to do it that way, fine. And I accept Genesis as a, in its historical context, and that's quite happy with all that. But I still have one thing that bugs me a bit, and that is the centrality of Adam in the story as, let's call it, the first man. Now, somewhere along the line there has to be an Adam. In other words, if you have the gradualist theory, let's say, it's maybe a little bit out of our brief, I don't know, but we talk about a soul. Man has a soul, and it's sort of his lifeline is God, or however you like to look at it. I don't know if animals have souls or not, no idea, but with the Genesis account, biblical account, it's all nice and cut and dried. There is Adam, first man, and so on. Now with the gradualism concept, which from a scientific point of view I'm extremely happy with, an animal gradually turns into a man, putting it perhaps a little bit crudely. What do we do about, what's our concept of where the soul comes into the story? I don't know if that's a fair question to ask you or not, really.
Sure. I mean, it's something I
Everybody's
think
got
about all
a
the
view on
time. It's among the most important questions that one can ever ask. So, from the biological point of view, I would suggest that one possibility is that as Scripture speaks in terms of God breathing into Adam the breath of life, that he was breathing in spiritual, this is one possibility, breathing into particular being at that point, the spiritual breath of life, and humankind at that point became living creatures spiritually, and so something unique about God's entering into relationship with a couple that then became the beginning of God's working with people down through history. So there's a biologist, not a theologian, giving one example of the sort of thing that I do as a biologist as I think about this. Certainly interacting with the theologians within my own department at a Christian university, evangelical university in the States, a number of them would, probably most of them would not agree with that particular interpretation. They would have another interpretation. Most of them would indicate a belief of the notion that Adam and Eve are representing us, and that it's not meant to be historical. It's the story of the ages, and indeed all of us go our own way, and that story needed to be told in a way that would last on through the generations. And it's our story that indeed God makes us perfect, but we choose to go our own way. And God, all of the things in the story are symbolic in many ways, regardless of whether it's history or whether or not it's purely theological. So, for example, and there's people who can talk about this more eloquently than what I can, but I'll just summarize it quickly. And if anybody wants to jump in, they can. But so, for example, it could well be that it is that the statement about being naked and knowing no shame. Of course, that's what God intends for us, and that's the way we are at first. And then we choose to go our own way, and we, in a sense, spend our lives trying to cover up for that apparent nakedness that we feel exposed because we know we're not right with God. We know we're not where we should be with God. And the notion of God being able to speak, us being able to commune with God, that's what God wants for us. And that's what was in the garden. And then the guilt that was associated with that, having to go and hide. Other parts of the story relate to the relationship between a man and a woman. Of course, Jesus refers to that one flesh relationship. And so all of that, no matter what, has tremendously important theological messages to us. And so some people would be very comfortable with that just simply being our story. Other people, because they really want it, for them it's important to be historical, then there's the other approach, and that is that it really is a real couple. And it's the beginning of a relationship with God that began with that real couple.
That's the first hand I saw. Go up, and then we'll...
I'll maybe show my ignorance here. In our study of Genesis earlier, there was this stress on not interbreeding. What's the sort of genetic implications? in the genetic code, what stops breeding between species, especially those that you maintain have a common ancestor?
So, from the biological point of view, which is now what you're asking, from the biological point of view, that's really the basis that you're really asking, the essence of speciation in general, which of course is the big question. Darwin himself wrestled with that question of speciation, even though he didn't write about it hardly at all in Origin of Species, ironically. His notebooks show that he had some very mature ideas of the sort that people were still working through over 100 years later. The significance of geographical separation, for example. So you have a group of organisms in one geographical region separated from a group of organisms in another geographical region, and thereby gradually changing in a way that reproductively isolates them from this group over here because of new mutations that have occurred that result in them not being able to crossbreed. So there's been a lot of work on that question, origin of species, especially over the last 50 years.
So does that mean there would have to be two of these apes to have that same chromosomal change in order for them to breed?
Breeding is a physical thing, isn't it? Mostly it's just physically whether they can have sex.
Well, that was my question.
So you don't get it doesn't happen overnight. It's a process, and so you would get maybe isolation of a small population of that somehow gets isolated. This is just one example of the sort of thing that people believe is important, and so gradually, so it's not like overnight you get a new species. It's a process of isolated populations. That's one of the ways in which this really took her.
And eventually their chromosomes just become incompatible, even as similar as we are to the chimps, because of the outworkings of the genetic differences. So you wouldn't get a chimp through the fact.
I think the next one is you.
You started a sentence which I thought was going to contain the answer to many of our prayers when you said the answer to pain and suffering is between the two flavours of God. And I failed to capture adequately these two flavours. You talked about the spiritual flavour and the chaotic flavour. Could you just go back over that, please?
Freedom and necessity, it was in there someplace. Freedom, so God's will, or I think it was freedom and sovereignty at that point when I used that. And so freedom, meaning that God's will, as John Polkinghorne spoke of today, so God willed that that creation have a free component, but then still, we have God sovereign, and people. So God is sovereign, and the answer to that someplace is that it's between the freedom that God wills for us, but then also the fact that God is sovereign and in control of the whole thing. And maybe some other people might want to jump in on that, because I'm a biologist.
Yes, I think your sentence opened up all sorts of questions which we get asked.
Sure.
As a hospital chaplain. 2 years ago, was it? Everybody asked me, after the Christmas break, why was the tsunami? And I was at the bedside saying, I do not have an answer to this. And then, you go to the bed of somebody with severe cancer and they say, don't tell me about the tsunami, tell me why God has given me this incredible pain. I don't have an answer to that one either.
And God didn't answer Job, and that's that's a point I especially want to make. And so sometimes I feel as though when we try, and Job he worked so hard to try to sort it out. The same kind of thing that Jesus was asked in John chapter 9. Somebody, it might have been you that was asking that question last night, where the parents came in and they said, people came by and said, who is responsible for this man being born blind? Was it his father or his son or his mother's lineage that was responsible for this happening? So here was Jesus' opportunity to answer that very question. Why is this happening?
Yes.
And of course we know what Jesus did. He just healed me. That was
said
Jesus'
so that
answer.
God's glory might be shown.
Yes.
That was his answer. I'm just writing a book on natural disasters.
Please
in
do.
about 2 years' time.
Please do.
James is next.
I actually had a question along the same lines as your talk about freedom and necessity and chance in creation. My question is, how does freedom relate to chance? Because my understanding of the Christian tradition of reflecting on freedom is that freedom is always based in rationality and understanding. So a dripping tap may be chaotic, but it's not free. So how do you associate, what is your basis for putting a kind of understanding of evil in the world as God making the world free, at the same time as saying that this freedom is somehow related to chaos? How does this freedom and chaos relate?
I don't really see the problem that you do, I don't think. To me, freedom means free for randomness. And I don't see that there's a problem in being free for randomness to work itself out.
Okay. I suppose I'm thinking the freedom that God has given to humanity, which is the freedom the Bible usually talks about, is freedom to follow intention, or freedom to follow will, to have some kind of effect in that way. And when you talk about, and I suppose in some sense an impersonal nature having freedom, those two concepts don't make sense to me. I don't understand a chaotic
they certainly
thing.
play themselves out, for example, in the human body.
Yeah.
Where that freedom, in a sense of God not intervening, for example, in the case of cancer. And so we have these random events, these mutations that take place within the body. And it, in essence, creates chaos. And isn't that a result of God granting freedom?
I wouldn't say
in that
that's
general
freedom.
sense, including the cells.
Yeah, I wouldn't say that's freedom, though. I'd say that's chaos. Just like in Job that you talked about, God doesn't say, these things happen, these bad things happen because there is a necessary element of chaos in the world that I like. He says, he doesn't say, this isn't my responsibility. He says, okay, maybe I did do all this, but I'm in charge. I get to. And maybe you don't understand the good purpose that I have. But that's because of your limitation, not because I'm not within my rights. So I'm just confused about the whole, chaos theodicy thing, I suppose.
Well, and of course, the answer to the question has never really been answered, and so we all would like to know that same answer that Job had, that Job did not get an answer to either. I don't know. Anybody else want to make any comments on that?
Well, Job chapter 1 actually gives us another perspective again, because there we see what Job never sees,
Right.
is that Satan comes to God and says, does Job fear God for nothing?
Yes.
And so the way that plays out ultimately is God drawing out of Job the faith that ultimately triumphs over everything that is going on. And it is a faith that is willing to trust God even though he cannot see, even though he slay me, yet will I trust him.
Yes. Yes.
And that's why I think your use of Job 42, your use of Genesis 50 is so important in this whole discussion, because there are things that we won't know this side of heaven, but we trust in a God who, despite the chaos, the apparent chaos, we still know is in control and is a good God.
And it's so interesting because God could have told Job there at the end, okay, Job, here's this story. I was sitting down with Satan. I had this conversation with Satan, but he doesn't. And the reason, I think, in my opinion, the reason Job is written that way is that God wants us to know that there are things we're not going to be able to understand.
So the answer is that the interpretation is then that things appear random, but they're not actually. God is in control, and we just don't know how that's working itself out.
No, I wouldn't agree with that. But I might have to think about it a little bit more before I would want to say too much more.
Sorry for...
Very interesting point. Thanks so much.
I think we'll have to have one last question, then a break, and there'll be lots more chance this evening. But actually, I will just make I'll make one comment about the natural disasters. That actually, as I've done research on it, it is probably the case that something like 80 or 90%, maybe even more, of the fatalities are avoidable. And they were in the tsunami. They were in lots of earthquakes and other examples. And not got time to talk about why now. But in that sense, they are generically the result of a fall in humanity. Humanity has the know-how to prevent those things happening, and it hasn't. So in that sense, it's a result of the fall, a result of sin. Now, it doesn't explain all of the fatalities. There are some which, as far as we can tell, were unavoidable. But
if I could just add something to that.
This mystery. Yeah.
If I could just add something to that, because I think that's a really interesting point. In the case of Jesus, however, he had that opportunity to say it was sin that was responsible for this. God, with respect to Job kept saying, Job's friends kept saying it's sin, and the answer in the end was it's not sin. And so, although 80 to 90%, but still there's that other 10 or 20%.
Indeed. There's still an issue. Yeah.
For many Christians, evolution is seen as an attack upon Christian ideas and values, and I think many Christians who believe in evolution sort of have to fight back to say, well, it hasn't done that, it's safeguarded. But can you actually go further than that and say, actually, evolution has given me some insights into God, which are very helpful, rather than we're just defending the ground, we're going beyond that. No, actually, this teaches us things about God which are beyond the traditional understanding of creation.
From my perspective, the story of life as a biologist mirrors the story of our lives as believers, where the hand of God is always working through creation in ways that we can't necessarily decipher, but we can look through the products, and so on, we can look at it. And so I love that unity that's associated with God working in much the same way in life as God seems to be working in the Bible, as God seems to be working in our own lives. And so, for me, that's very satisfying.
Richard, did you want to make a last comment? Sorry, I've seen you.
Yeah, I just, in responding to the issue of Job, I really think that Job is the great existential book because it's really more of a search of Job for the presence of God. And it's not really even Job asking so much, why am I suffering, but he's asking, where is God in the midst of the suffering? And in the end, the response of God is to take Job out of the center of the picture and say that, look at all I've created, look at the greatness of it, and look at what can't be understood. And you're not going to understand it. And there is this kind of removal of the concerns of Job as being the center of the universe, and saying it's not that they're illegitimate, it's just that they are not part of, and they're not going to be answered in this life.
Yeah, I think you said that so well, Rick. I appreciate that.
Well, thank you very much. I think we'll have a 10-minute break now to stretch our legs and so on before John comes and gives us the last talk. So let's Darrel get going.